Hi, everybody. Welcome back to week 2 of your Advanced Patho for Advanced Nursing Practice class. Again, I'm Jessica Lanfranchi. I'm your professor for this class. And again, I'm so grateful and happy to be guiding you in this particular class to hopefully really give you a strong foundation in patho. So week 2, here we are. So in week 2, we're going to be discussing molecular biology, genetics, and genetic diseases, as well as cellular physiology and pathophysiology. I have incorporated some videos in this particular PowerPoint to really give you some visual explanations to help ground some of this information for you, because I know the textbook can be quite overwhelming as it gets into the details and the nitty-gritty of biology and cells, especially at the cellular level of the body. It's a lot. So I hope that these videos will help in grasping some of these concepts. This particular week, our assigned readings were for Chapter 3 and Chapter 4. Okay, so before we move on into this week's modules and readings, I wanted to just review week 1. Week 1 included the foundational concepts of clinical practice, understanding the role of molecular and biologic disease, and interpreting altered cellular biology. In week 1, we talked about the foundational concepts of clinical practice. And in Chapter 2, we talked about the chemical and biochemical foundations. In Chapter 1, it really gave you kind of the roadmap of this particular textbook, Anatomy, Physiology, and Patho. It kind of mapped out like the rationale for the book structure. It started with a review of the first couple of chapters in terms of like just talking about the basics of science. That's like chapters 2 through 4 of the book, followed by the topics of infectious organisms and the organ system chapters, which is basically chapters 5 through 17. And then in chapters 5 through 17, it aims to focus on major principles and components of underlying normal function of the organs and systems, with each principle or component immediately followed by discussions of the disease resulting from the specific pathophysiological alterations in that particular principle or component. I mentioned to you last week in Chapter 1, like I would definitely suggest in terms of your note taking, you use Chapter 1 as kind of an outline to guide your notes throughout your textbook. So the big thing that we really wanted you to look at this chapter was just to give you like a preview of the major principles and components that are focused on in each chapter throughout the book. Okay. So then Chapter 2, we get into the chemical and biochemical foundations. And so here we really, basically in this chapter, we really talk about the fundamental, foundational and reviewed information from basically your undergraduate science prerequisite courses. So a lot of it was kind of review, but most of it was discussing the chemical and biochemical foundations of life. They started with reviewing chemical foundations, including like the atomic structure and the periodic table of elements, which we will review again before I move on into our coursework for this week. Focus is placed on the most common elements found in the body and how electron shell filling impacts their chemical reactivity. It talked about the formation and role of ions, especially acids and bases within the body. And the text went into describing how the role of the ions. And it also talked about the importance of electron sharing and formation of polar and non-polar covalent bonds. It talked about the application of polar and non-polar bonds and molecular structures to the properties of biomolecules. And it really went into their significance to human physiology. They focused on the role of carbon as the foundational atom of life, and they really got into the details of that. And they went into the 4 specific classes of molecules, their structure, and some of their body function. And that we will also get into greater detail in this particular module this week. But they went into the 4 classes, which They really discuss more of the greater detail, the carbohydrates, lipids, amino acids, proteins, and nucleotides. So I'm going to go through that in my next slide. Let me just show you. Oh, and before I forget, so the other thing I did this week too, and I incorporated into my PowerPoint, was the thought questions. I went through your chapter 2 and chapter 3, which we'll go through in this review really quickly. I want to make sure that you use those questions to help guide your reading. So that's super important. Okay, so page 3. So here we have, I actually listed the page numbers for all of the thought questions. So if you want to follow along in your textbook, I would highly recommend that, just so you can really get a visual of where the particular subject matter applies to your readings, because it gets a little confusing in that book, I think, if you're not careful. Okay. So page 12 in your text, what are the major and minor elements that make up the body? Okay, super easy. Major elements are your carbon, oxygen, hydrogen, and nitrogen, and the minor elements are your sodium, potassium, chlorine, calcium, phosphorus, sulfur, magnesium, iron, copper, and zinc. Okay, how many electrons are in the outer shell of the major elements, or valence, I should say? Just keep in mind the first shell can hold 2 electrons, and the second and third shell both can hold 8 electrons. Hydrogen has 1 electron in the first shell. Carbon has 4 electrons in the second shell, and nitrogen has 5 electrons in the second shell as well, while oxygen has 6 electrons in the second shell. Number 3, how many members of group IVA4A are in the group of elements that make up the body? Carbon is the only element in the body that is in group IVA4A and having 4 electrons in its outer shell. Number 4, what are the ways in which the duet or octet rule can be satisfied for an atom with an incomplete valence shell? Keep in mind, the octet rule means that there are 8 electrons in the outer valence shell. In order to satisfy the octet rule, electrons may be lost or acquired. This process results in an ion. Acquiring an electron forms an anion, a negatively charged ion, while losing an electron forms a cation, a positively charged ion. Ions are stable in the aqueous intracellular extracellular fluids as their electrical charges are surrounded by polar wet molecules. The other way of satisfying the octet rule is by electron sharing between atoms in the process of covalent bond formation. That's a lot. Number 5, what are the main ions found in the body and where are they found? Are they in the extracellular fluid or intracellular fluid? Okay. So most ions in the body are dissolved in aqueous intracellular, intracellular and extracellular fluids. The main ions in the body are sodium, chloride, bicarbonate, potassium, phosphate, hydrogen, magnesium, and calcium ions. Several amino acids found in proteins can also be ionized, and soluble proteins found in the intracellular and extracellular fluids are often negatively charged. Okay, so hopefully these thought questions were something that will help solidify what you have done in terms of your readings. So again, I just wanted to give you a little bit of visual in terms of the major and minor elements that make up the body. So here we have our periodic table. In green, you have your common elements, and in purple, in purple you have your trace elements, and in pink you have your remaining elements. Okay. Oops, sorry guys. Okay. So again, I just like these graphs because they really focus in on giving you an idea of the percent of total body weight in the body of the elements. So here we really can see, the major elements are carbon with 18.5% of your total body weight, oxygen obviously at 65, hydrogen at 9.5, and nitrogen at 3.2. Those are your highest, most major elements in your body. Okay, So ions versus atoms. So I just wanted to just focus in on the difference between an atom and an ion. Remember, atoms are electrically neutral, where ions are electrically charged, okay? Atoms have the same number of protons and electrons. Ions, on the other hand, have a different number of protons and electrons. And an atom, the number of protons on the atom defines its elements, whereas an ion may contain one or more atoms. Now, what are ions? Ions are atoms that contain an uneven number of proton and electrons, which results in an overall positive or negative charge. So cations are atoms that contain a positive charge, which means that they contain more proteins than electrons, versus an anion, which is an atom that contains a negative charge, which means that it has more electrons than protons. Okay. So another big thing I wanted to just make sure we reviewed, and I wanted to show you a short video just to really reinforce it, was about PKU. So let's go to this video. Oops, I wonder if it'll let me play it. Sorry. Here we go.
Phenylketonuria, or PKU, is a rare metabolic disease that can lead to severe brain disorders caused by the accumulation of the amino acid phenylalanine to toxic levels in the blood and brain. Amino acids are the basic building blocks that make up proteins. Phenylalanine, or Phe, is one of the essential amino acids. It's necessary for life, but our bodies can't make it. So, phenylalanine must be acquired through foods that contain protein. Normally, once phenylalanine enters the body, most of it is converted to tyrosine by the enzyme phenylalanine hydroxylase. Tyrosine is then turned into neurotransmitters important for normal brain development and function. PKU is an autosomal recessive genetic disorder that affects function of the phenylalanine hydroxylase enzyme. The phenylalanine hydroxylase gene is located on chromosome 12, and over 600 mutations have been described. The degree of enzyme function can vary. When untreated, people with PKU develop symptoms such as severe intellectual disability, psychiatric disorders, and seizures. A pregnant woman with PKU must pay special attention to her phenylalanine levels to reduce the risk of maternal PKU syndrome that can result in heart defects, microcephaly, and developmental disability in her baby. In PKU, elevated phenylalanine levels and reduced tyrosine levels can change the way the brain functions. This is because phenylalanine uses the same transporters to get across the blood-brain barrier as other amino acids, including tyrosine and tryptophan. Tyrosine is needed to synthesize dopamine and norepinephrine, and tryptophan is needed to synthesize the neurotransmitter serotonin. As phenylalanine levels rise, it occupies all transporters, making it hard for tyrosine and tryptophan to get across the blood-brain barrier. As a result, dopamine, norepinephrine, and serotonin levels in the brain begin to fall, leading to abnormal brain development and intellectual disability. Testing for PKU is typically done as part of routine newborn screening, approximately 24 hours after birth. Screening is done by taking a small blood sample from a heel stick and measuring blood phenylalanine levels by a simple laboratory test. If the screening test shows high levels of phenylalanine and low blood tyrosine, a different method is used to repeat the measurement of the blood amino acids and to confirm the diagnosis. First-line therapy for PKU is the low phenylalanine diet. This includes medical foods, such as low or no phenylalanine medical formulas that are a synthetic form of protein, and foods modified to be low in protein. All high-protein foods, such as meat, fish, eggs, and dairy, are eliminated from the diet. Measured amount of some grains, vegetables, and fruits are allowed, depending on the individual patient's tolerance of small amounts of phenylalanine. Each patient will need to customize the amount of phenylalanine dietary intake with medical professionals according to their individual needs. In addition to the low phenylalanine diet, 2 FDA-approved pharmaceutical treatments are now available. Kuvan is a cofactor for phenylalanine hydroxylase, which helps improve the PKU patient's innate PAH enzyme activity and increase their phenylalanine tolerance. Kuvan must be used in conjunction with the low phenylalanine diet. Palynziq can be used in adults with PKU. Palynziq is an enzyme injected under the skin every day by the patient that circulates in the blood, metabolizing phenylalanine, and substitutes for the defective phenylalanine hydroxylase enzyme. Palynziq works well for many adult PKU patients, but it can have serious side effects in some patients. All right, as a quick recap, in PKU, phenylalanine levels build up and tyrosine levels drop. Symptoms in untreated individuals include seizures and severe developmental delay. In the US and much of the developed world, newborns are routinely screened for PKU. Treatment, initiated soon after birth, is based on a diet low in phenylalanine that requires the use of an amino acid-based medical food or formula, severe restriction of dietary natural protein intake, and the ingestion of modified foods that are lower in protein. 2 FDA-approved drugs are also available for the treatment of PKU.
Okay, great. I hope this was helpful just to recap on PKU.
It's 5 o'clock.
All right, so let's finish our thought questions. So page 18, number 6, what are the similarities and differences between iconic, nonpolar, and polar molecules? Iconic molecules are those in which one or more atoms has lost or gained an electron to have a full charge of 1 plus, 2 plus, 3 plus, or 1 negative, 2 negative, 3 negative. These ions will attract other molecules and single atom ions of opposite charge and are stabilized when surrounded by polar water molecules in aqueous solutions. Nonpolar molecules are those in which there is equal electron sharing across covalent bonds. When a molecule is made up of elements with differing electronegativity, partial charges develop within the molecule. Remember, the strength of attraction on electrons is shared in a covalent bond. The element with stronger electronegativity develops a partial negative charge, and the element with lesser electronegativity develops a partial positive charge. Focusing on biological molecules, those rich in carbon-hydrogen bonds, such as fatty acid chains or based on cholesterol molecules, tend to have relatively equal electron sharing and are nonpolar. On the other hand, carbohydrates, amino acids, and nucleic acids containing oxygen-hydrogen bonds and nitrogen-hydrogen bonds tend to have unequal electron sharing and to be polar molecules. Now, number 7, can you draw and label a water molecule showing the special properties that make it an excellent solvent for body solutions? So the most important thing about a water molecule, as you see in this diagram, is that it has a polar bond between the oxygen and the hydrogen atoms with a partial negative charge on the oxygen and a partial positive charge on each hydrogen. Each molecule also can make up for up to 4 hydrogen bonds with neighboring water molecules, which is what is responsible for surface tension and the tendency of water molecules to stick together. Hydrophilic solutes can dissolve in water as water molecules surround polar molecules and ions. Keep in mind, water dissolves more substances than any other liquid. And the bent molecule shape, it makes it polar, which it gives the oxygen atom a partial positive charge and the hydrogen atoms partial positive charges as well. Okay, good. I hope these were helpful. Okay, number 8. What features of carbohydrate molecules contribute to their tendency to be polar and hydrophilic? Carbohydrate molecules contain multiple polar hydroxyl groups that can easily form hydrogen bonds in water and aqueous solutions. And then how many biological functions of carbohydrates can you identify? We're going to get more into that in just a minute. Biological functions of carbohydrates include energy source for cells, modification of cell membrane proteins, modification of plasma proteins and hormones, cell recognition sites, attachment sites, and support within cell membranes, as well as mediation of host and pathogen interactions. Can you identify 3 lipoproteins and identify major function that each type serves in the body? And why might a person need to have a laboratory assay to determine their lipoprotein levels? So the answer to these two, let me show you. So here we go. First, let's just talk about the functions of carbs, just as we had listed before. They are a source of energy, they're storage of energy, their structural component is found in plant cell walls. Their structural component is found in DNA and RNA. And they also have a structural component found in glycoproteins and glycolipids, which attaches to cell membrane proteins and phospholipids. And here are your different kinds of carbohydrates. And here's your lipoproteins. So the three lipoproteins that we could identify are chylomicron, which carries products of dietary fat digestion from the intestine to the liver and other organs. Very low density lipoprotein, which carries, they're called VLDL, which carries newly synthesized triglycerides from the liver to the adipose tissue, as well as to other organs and tissues. We have IDL, which is intermediate density lipoprotein, which is your IDL, which is transient lipoprotein formed as tissues remove triglycerides from the VLDL. Then, of course, we have the low density lipoprotein, which transports cholesterol throughout the body. And then we have our high density lipoprotein, which is formed in the liver and intestines and transport cholesterol from the peripheral tissues back to the liver. So this little diagram here on the right gives you a nice visual picture of what lipoproteins do. And why are the lipoproteins required for transport of lipids throughout the body? So lipids, such as cholesterol and triglycerides, are nonpolar hydrophobic molecules and are not soluble in aqueous solutions. So therefore, the lipoprotein particles carry the cholesterol and triglycerides in a hydrophobic core that is coated with phospholipids and proteins that can form hydrogen bonds with the aqueous plasma to travel throughout the body. And why would a patient need to have a laboratory assay to determine their lipoprotein levels? Well, obviously we know lipid panels are assessed as a marker for cardiovascular risk, right? So having an excessive amount of the low density lipoproteins, or AKA the LDLs, having excessive amounts of them can, so having excessive amounts of LDLs can migrate into arterial walls and promote atherogenesis, and that causes atherosclerosis. Higher levels of HDL are generally associated with lower cardiovascular risk. So remember, your HDL is your good cholesterol. Now, let's keep going here. All right, so your thought questions. All right, so these are the last of it. Can you name 3 different amino acids that are associated with specific body functions and those functions? So the 3 I listed here are tyrosine, what else do we have? Tyrosine, tryptophan, and histidine, glycine, and glutamine. So there's more than just 3. So keep in mind, amino acids are the building blocks for protein synthesis. However, some individual amino acids also have specific functions within the body. Tyrosine, for instance, is used in the synthesis of thyroid hormones, as well as neural and endocrine catecholamines, like dopamine, norepinephrine, and epinephrine. Tryptophan is used to synthesize serotonin, melatonin, and adenine dinucleotide, which are used in metabolic functions. And histidine is used to synthesize histamine, an inflammatory mediator. Glycine is used in the production of porphyrins, which are precursors to heme. And glutamate is a neurotransmitter and is the precursor of GABA and another neurotransmitter. Okay. What are some, how does hydrogen bonding contribute to secondary protein structures? Hydrogen bonds between the carbonyl and amino portions of peptide bonds stabilize the secondary structures of alpha helices and beta plated sheets within proteins. Okay, what are some hydrophobic amino acids that might be found in the core of a folded protein? So protein folding tends to cluster hydrophobic amino acids in the core of the protein, hidden away from the aqueous extracellular or intracellular fluid. These include alanine, valine, leucine, isoleucine, methionine, proline, tryptophan, and phenylalanine. What are some hydrophilic amino acids that might be found on the outside of a folded protein? So hydrophilic amino acids are usually found on the outside of proteins facing the aqueous solution of intra and extracellular fluids. These include polar amino acids, asparagine, glutamine, cysteine, serine, as well as charged amino acids, aspartic acid, glutamic acid, lysine, arginine, and histidine. Okay, and what are some of the roles individual nucleotides play in cell function? So ATP, or adenosine triphosphate, is the energy currency of the body, ATP, very important. The cyclic forms of adenosine monophosphate, or CAMP, and CGMP are intracellular second messengers. GTP, or guanosine triphosphate, is involved in signal transduction by membrane receptors. And finally, number 18, which DNA base pairs participate in complementary base pairing through hydrogen bond formation? Adenine and thymine form a base pair and are connected by 2 hydrogen bonds. Guanine and cytosine form a base pair and are connected by 3 hydrogen bonds. Okay. Okay, so amino acids. So amino acids, I just wanted to really review them again from last week. Remember, they are the building blocks of protein. They're huge in the pathophysiology of the body. So there's 20 kinds of amino acids, okay, free amino acids, I should say. And we have essential amino acids and non-essential amino acids. And the essential amino acids are obtained from your nutrition, and your non-essential amino acids are synthesized by the body. And they have such a role in your body. So look at all these things that the amino acids does. It does protein synthesis, cell signaling, it does acid-base balance, it affects your appetite, your body composition, your blood flow, growth and development, immunity and health, ammonia removal, metabolic regulation, reproduction, stress response. I mean, there's a lot of stuff, RNA and DNA synthesis. All of these things are what, the functions of the amino acids. Okay, so remember, amino acids are the building blocks of protein. So keep in mind that proteins provide extremely essential functions in the body. Obviously, the antibodies support immune function. They provide support to the body. They support muscle contraction and movement, they move essential molecules around the body. They support the regulation and expression of DNA and RNA. And they help digestive enzymes help facilitate chemical reactions. So what is the protein made of? The 20 amino acids form the building blocks of proteins, of which 10 are essential amino acids. So what is protein? They're long, proteins and amino acids go hand in hand. They're long chain-like molecules made up of amino acids. So proteins grow, maintain, and replace the tissues in our bodies. There are 20 different amino acids. 8 of the 20 amino acids cannot be produced by the body and are therefore called essential amino acids that are obtained by eating certain protein-rich foods. So it's important to know this because now we get into the point of nutrition. So keep in mind that too much animal protein can be dangerous to the body as it causes a strain on the liver and kidneys, causing them to work extra hard to get rid of the extra protein, which is not the case in plant protein, but that's a whole different story. So why focus on protein? Because, we need this to keep strong, heart healthy diet is, filled with high quality lean protein, which helps reduce the risk of chronic disease and obviously the reduction of blood pressure. So amino acids in real life, like what does this all have to do with your actual practice as an NP? So in order to understand like the importance of the amino acids and apply the pathophysiology, you need to understand like what the amino acids are and why this is so important in terms of nutrition. The amino acids help build the muscles. So without these, we're in trouble, right? And where do we get the amino acids in terms of food? Here are your main food sources: oatmeal, nut butters, hemp seeds, beans, quinoa, chia seeds, nuts, broccoli, and spirulina. These are all actually examples of plant-based or vegan sources of amino acids. But over here, we also have other sources: eggs, peanuts, whitefish, tofu. These are all different sources of your different types of amino acids, depending on what you need. But a lot of it comes from eggs. So just keep that in mind. Chia seeds are another excellent source of amino acids as well as fiber. Okay, now let's get into week 2. Let's get focused. So in week 2, we're discussing, we're getting into genes and genetic disease, the exploration of how lifestyle impacts disease and disease states, recognizing how environment influences genes, and exploring how stress impacts health and disease. So here we have a normal gene versus a mutated gene, which has no protein. We are getting into the cellular level of genes and genetics. So brace yourself. Okay, so this week we really got into chapter 3 and chapter 4 of our text. Chapter 3 being molecular biology, genetics, and genetic diseases. Chapter 4 being cell physiology and pathophysiology. In chapter 3, it really focuses on genetic disorders and how are diseases inherited. So in order to understand the answer to that question, you really have to understand how genes are passed through generations and an understanding of how the mutations work both in Mendelian genetics and non-Mendelian genetics. Current research studies are still revealing many ways in which genetic variability occurs and all the variables that can contribute to varying phenotypes. The future of treatment of disease is also tied very closely to genetic research, as is our understanding of the disease process itself. So really in this chapter, you really are provided with a basic understanding of genetic, genomic, and epigenetic concepts. And then in chapter 4, we get into cell physiology and pathophysiology. So keep in mind, cells in the body perform so many different functions. The focus of this chapter is the physiology and the pathophysiology of cell functioning. We want to look at, in this chapter, we definitely looked at how substances cross the cell membrane, along with how these substances actually affect the cell. Cell signaling is described along with how cells pass messages among one another. Contractions of the types of muscle cells is reviewed. Cell renewal is covered, along with the impact of stress on the cellular level. We talk about cell death as a protective mechanism, but can also be a pathological process. And then at the end of the chapter, they get into the gerontological considerations of aging, including frailty and the effects of inflammation. Okay. So, here. Okay, so factors that affect genes. So I love this little chart. I think it is such a great little standardized way of just kind of pointing out factors that affect genes. And it's evidence-based. You see it's based on cohort studies. We like evidence-based practice. And so this just gives us really a basic outline of how it works. So unhealthy diet, sedentary lifestyle, high stress, medications, poor sleep, addiction, social isolation. And then, of course, you have microbiome alterations, epigenetic changes, and cellular stress injury, which will lead to inflammation, which then leads to disease processes, right? So all of these affect our genes at a cellular level. And how do we change any of this, or what can affect it? Lifestyle interventions, of course, and behavioral changes. This is the website for this particular study and article, and it's really very interesting. So if you wanted to learn more about this particular part of this chapter, I would suggest you go to this website. Okay, so we get into DNA in this chapter, and we really look at DNA as a molecule. So keep in mind, DNA is the molecule of genetic inheritance. Germ cells, aka egg and sperm, each contain 23 chromosomes, 22 autosomes, and 1 sex chromosome. Okay. Upon fertilization, the resulting zygote has the normal human chromosome content, consisting of pairs of homologous autosomes, 1 to 22, and 2 sex chromosomes, either XX or XY, for a total of 46 chromosomes. Okay. DNA is found in the cell nucleus and is made up of long polymers of nucleotides. Each polymer strand is connected to a complementary strand through hydrogen bonds formed between opposite bases. There are 4 DNA bases, okay? And there you can see them listed here: adenine, cytosine, guanine, and thymine, okay? These complementary base pairing joins A to T pairs with 2 hydrogen bonds and G to C pairs with 3 hydrogen bonds. Okay. Okay. So here we get into epigenetics. And so epigenetics is the field studying non-mutational influences on gene expression and disease mutations. I really wanted you to get a better understanding of what exactly epigenetics is, so I wanted to play a short video just to kind of go over the basis of it.
Here's a conundrum. Identical twins originate from the same DNA, so how can they turn out so different, even in traits that have a significant genetic component? For instance, why might one twin get heart disease at 55 while her sister runs marathons in perfect health? Nature versus nurture has a lot to do with it, but a deeper related answer can be found within something called epigenetics. That's the study of how DNA interacts with the multitude of smaller molecules found within cells, which can activate and deactivate genes. If you think of DNA as a recipe book, those molecules are largely what determine what gets cooked when. They aren't making any conscious choices themselves. Rather, their presence and concentration within cells makes the difference. So how does that work? Genes in DNA are expressed when they're read and transcribed into RNA, which is translated into proteins by structures called ribosomes. And proteins are much of what determines a cell's characteristics and function. Epigenetic changes can boost or interfere with the transcription of specific genes. The most common way interference happens is that DNA, or the proteins it's wrapped around, gets labeled with small chemical tags. The set of all of the chemical tags that are attached to the genome of a given cell is called the epigenome. Some of these, like a methyl group, inhibit gene expression by derailing the cellular transcription machinery or causing the DNA to coil more tightly, making it inaccessible. The gene is still there, but it's silent. Boosting transcription is essentially the opposite. Some chemical tags will unwind the DNA, making it easier to transcribe, which ramps up production of the associated protein. Epigenetic changes can survive cell division, which means that they could affect an organism for its entire life. Sometimes that's a good thing. Epigenetic changes are part of normal development. The cells in an embryo start with one master genome. As the cells divide, some genes are activated and others inhibited. Over time, through this epigenetic reprogramming, some cells develop into heart cells and others into liver cells. Each of the approximately 200 cell types in your body has essentially the same genome, but its own distinct epigenome. The epigenome also mediates a lifelong dialogue between genes and the environment. The chemical tags that turn genes on and off can be influenced by factors including diet, chemical exposure, and medication. The resulting epigenetic changes can eventually lead to disease if, for example, they turn off a gene that makes a tumor-suppressing protein. Environmentally induced epigenetic changes are part of the reason why genetically identical twins can grow up to have very different lives. As twins get older, their epigenomes diverge, affecting the way they age and their susceptibility to disease. Even social experiences can cause epigenetic changes. In one famous experiment, when mother rats weren't attentive enough to their pups, genes in the babies that help them manage stress were methylated and turned off. And it might not stop with that generation. Most epigenetic marks are erased when egg and sperm cells are formed. But now, researchers think that some of those imprints survive, passing those epigenetic traits on to the next generation. Your mother's or your father's experiences as a child or choices as adults could actually shape your own epigenome. But even though epigenetic changes are sticky, they're not necessarily permanent. A balanced lifestyle that includes a healthy diet, exercise, and avoiding exposure to contaminants may, in the long run, create a healthy epigenome. It's an exciting time to be studying this. Scientists are just beginning to understand how epigenetics could explain mechanisms of human development and aging, as well as the origins of cancer, heart disease, mental illness, addiction, and many other conditions. Meanwhile, new genome editing techniques are making it much easier to identify which epigenetic changes really matter for health and disease. Once we understand how our epigenome influences us, we might be able to influence it too.
Okay, great. I really liked that video. I thought it was a great visual description of epigenetics. It's kind of a hard concept to understand, but the reality of how fragile our genes are is really well explained with epigenetics. So here we have like a little guide in this particular slide. One, things that early experiences altering gene expression and shape development. So external experiences such as stress, nutrition, and toxins spark signals between neurons. Neural signals launch production of gene regulatory proteins inside the cell. Then the gene regulatory proteins attract or repel enzymes that add or remove epigenetic markers. Now, number 4, epigenetic markers control where and how much protein is made by a gene, effectively turning a gene on or off, thereby shaping how brains and bodies develop. That's crazy. To me, that's crazy that our genes and our markers are so sensitive to our environment as well as what we're putting into our body. It just has to make us so much more aware as both human beings and as practitioners, the importance of taking care of ourselves both inside and out. Okay, so now we're going to get into genetic disorders and just review the common genetic disorders that and the defects that they cause. So genetic disorders are often detected in fetuses and neonates by prenatal and newborn screening. In other pediatric cases, sometimes abnormalities of physical features or abnormal function detected at birth and during childhood development will prompt your genetic testing. For disorders such as inborn errors of metabolism, early detection through screening and confirmation through advanced genetic testing can be life-saving or life-extended when treatments can be initiated early. All right. Okay, so here we have different types of genetic screening and tests. So for prenatal testing, they have amniocentesis or CVS for detecting chromosomal abnormalities like trisomy 21 or Down syndrome. Newborn screening includes sickle cell anemia and PKU. We just learned about the PKU where they do the heel stick, a very minute test to really help prevent major complications in life later on. We have carrier screening. We have heterozygote tests for cystic fibrosis, sickle cell anemia, and Tay-Sachs disease. We have an inherited predisposition. So in that case, we would look at breast and ovarian cancer gene BRCA 1 and 2, and also for Alzheimer's disease. So those are people who already have that history of those particular inherited genetic genes. For characterizing tumor types, we're looking at breast cancer variants, which may indicate a response to drug Herceptin. For a diagnosis of genetic disease to confirm physical exam, we're looking for inborn errors of metabolism, muscular dystrophies, DNA repair disorders, anemias, and Prader-Willi syndrome. And then, of course, pre-symptomatic testing for adult onset disorders, we would be looking at Huntington disease. Okay, so next, pediatric and reproductive considerations in genetics. So I just wanted to make sure that you're conscientious of these particular diseases that, and this genomic precision diagnostic algorithm they have here for you to use as a kind of a guide in terms of where during conception you would be looking at particular diseases or what testing would need to be done. So for instance, here prenatal, we're looking at parental diagnostic, family history, looking at, those tests would go further if it was a high-risk pregnancy. If the woman had any history of stillbirths, those are types of testing that we would start looking at, tests that we would want to start screening for. In postnatal, you're looking at children diagnostic with any kind of mental impairments, developmental delay, or genetic diseases. That's when you would start doing diagnostics on your children. And of course, in terms of couples, you want to make sure that you look at both couples' family history and if either of the parents are affected by any kind of genetic diseases. So carrier frequency by population and condition. Here's two very common genetic diseases, cystic fibrosis and beta thalassemia, as well as sickle cell. Three, actually, I'm sorry. And 1 in 65 African Americans has cystic fibrosis. 1 in 75 African Americans has beta thalassemia. And 1 in 10 to 12 African Americans has sickle cell. So keep in mind, and then you have your Mediterranean population, also a high incidence of all three of those particular genetic issues. Okay, so prenatal genetic testing. There are 5 types of prenatal genetic tests which can be used for screening the risk of a baby having a genetic disorder or for diagnosing such abnormalities. So for screening, in terms of prenatal genetic testing, it's non-invasive and it's offered to all women. They do blood work, maternal blood serum. They do prenatal ultrasounds and they do non-invasive prenatal testing. And those are of a low or no risk to the baby, the fetus. And then for diagnosing, to confirm or rule out genetic problems, and they can be invasive, and they're offered after abnormal screening tests or known risk factors are found. So like if the blood work over here came back abnormal, or they weren't seeing good proper development in your prenatal ultrasound, that's when we would go for further testing, such as an amniocentesis or the villus sampling. And there are high risks with sudden death with these particular tests. So you have to really make sure the mother and the father are aware and consent to these tests, depending on, the reasons for them. Okay, so let's review your chapter 3 thought questions. So number 1, why are spontaneously occurring sequence variations such as single nucleotide polymorphisms and mutations biologically useful? Well, evolution could only proceed through spontaneously occurring changes in DNA sequence. So we wouldn't continue growing as humans without the DNA spontaneously occurring. Or there's changes in the DNA. So some mutations have survival value to the organism, and these tend to be retained. For example, in location where malaria is common, the sickle cell mutation is also common. Red blood cells in a person with sickle cell trait are poor hosts for the malaria parasite. So the sickle cell mutation has been retained over several thousand years. For the genes involved in the sickle cell disease, cystic fibrosis and hereditary hemochromatosis, there are survival benefits of being heterozygous. However, as autosomal recessive traits, the individual who is homozygous ends up with the disorder. Okay, number 2. What steps lead from reading a DNA sequence coding for a protein, aka a gene, to production of the final protein? A promoter region of a DNA strand instructs RNA polymerase where to begin transcription. The RNA transcribes the DNA template strand until it reaches the terminator region, which tells it to stop. During post-transcriptional processing introns, the non-coding regions are removed from the pre-RNA. The exons, or the coding regions, are then spliced together, becoming mature RNA used for translation. During translation, the mRNA is read by the ribosome in 3 base codons. The transfer RNAs matching each codon in a sequence carry the amino acids to be added to the polypeptide chain, ultimately forming the final protein. Number 3, is it possible for a deletion mutation to also be a frame shift mutation? Could you diagram an example in which a deletion mutation would not be a frame shift mutation? Okay, so a deletion mutation is the removal of base pairs, changing the actual number of base pairs in a coding or non-coding DNA sequence. A frame shift mutation is deletion or insertion of one or more bases, not divisible by 3, that changes the reading frame for transcription. Therefore, a deletion mutation can be a frame shift mutation. The diagram of a deletion mutation that is not a frame shift mutation should demonstrate the deletion of base pairs in a number divisible by 3. In general, which inheritance pattern is associated with the degree showing the greatest number of affected individuals? An autosomal dominant shows the greatest number of affected individuals because only one copy of the abnormal gene is required for the phenotype to be present. Number 5, how might you modify your answer if you had additional information about gene penetrance in a given disorder? If the penetrance is less than 100%, then there are some individuals who have the disease-related genotype, but do not have the associated phenotype. Reduced penetrance may be due to mutation type, variations in gene expression of different alleles, epigenetic factors, gene-environment interaction, age, sex, modifier genes, copy number variants, and variability in related genes. How does the principle of complementary base pairing contribute to DNA sequence analysis using hybridization arrays? A gene chip holds small segments of DNA in an array on a slide. These segments are synthesized based on the known sequence of clinically relevant genes called probes. The chip is exposed to DNA from the patient, which is treated with enzymes to break it down into smaller segments. The patient's DNA hybridizes to the gene chip according to the complementary base sequence, and spots that do not hybridize do not contain a complementary strand. And number 7, how does epigenetic modification of DNA compare with mutation of DNA in terms of the process and molecules involved? So epigenetic modifications are heritable changes that do not involve mutation of DNA sequence. They are modifiable by environmental and interpersonal factors, such as environmental chemical exposure, drugs, aging, and diet. There is variability over the lifespan that is specific to the tissue of the individual. Okay, great. So how does all of this information apply to us clinically, right? So you're learning all these really intense, very fine details of genetics. What does this mean in real life? Okay, so you learn about all these things in terms of genetics and the importance of finding out what is your patient's family history, and you learn to recognize genetic red flags, okay, which indicate high risk for an inherited disorder. Here are some examples of red flags that should prompt a referral to a genetic specialist. Now, keep in mind, there are genetic specialists out there. You're not going to have to apply this information in your clinical practice at this level, but the important thing is you understand the basic concepts so you know what the severity of it all means. So a known genetic disorder in family or family history of disease in multiple generations of an affected maternal or paternal relatives. So we have 2 affected first-degree relatives or 3 or more affected maternal or paternal relatives. So this goes back to your genodiagram when you do your 3 generation history, medical history on your patient's family. And these are the things you want to recognize. Are 2 of their first-degree relatives affected by a genetic disorder? Or are there 3 or more affected maternal or paternal relatives? So is anybody on the mother's side of the family or father's side of the family, are there 3 or more people on either side of those families that have a genetic disorder? And then is there a moderate risk on both sides of the pedigree? So for example, 2 maternal relatives with cancer and 2 paternal relatives with heart disease. So if that's the case on your particular patient where 2 of their mother's relatives have cancer and 2 of their father's relatives have heart disease, you know in that particular instance, that patient needs to be screened for both cancer and heart disease and really talk about lifestyle changes to promote healthy daily life things to prevent those types of genetic disorders from starting to show symptoms in this particular patient's life. So here's another one: early onset of disease in first or second degree relative, including breast, ovarian, or endometrial cancers, especially if they're less than 50 years old, colon cancer, prostate cancer, stroke, coronary artery disease, type 2 diabetes, or dementia. So if any of these things come up in your patient's family history, especially if it's in a first or second degree relative, meaning it's their parents or their grandparents, you need to start screening as early as you can for those particular things. Again, you're looking at your United States Preventative Task Force guidelines to really make sure you're screening appropriately. But again, if you have a family history genetically, we know we have to start screening sooner than later. Obviously, if there's any sudden cardiac death, history of sudden cardiac death in your patient's family history, that, especially in an individual who was originally considered healthy, that's something you actually need to screen your patient for earlier than later. And then, of course, ethnic predisposition to certain genetic disorders includes sickle cell, especially in the African Americans, Tay-Sachs disease, especially in the Jewish families, and alpha thalassemia, especially in people from Southeast Asia. That's really important, these three particular ethnic predispositions in terms of genetics. I will say, if you're going to get a question on genetics on your boards, the likelihood it will be something around this particular slide, whether it's ethnic predisposition, early onset disease in first or second degree relatives, you have to start doing earlier screening. And then, of course, if you have a known genetic disorder in a family, especially if it's in the first degree relatives or in 3 or more people within the family. So super important, guys, because this could, early intervention could definitely save your patient's life or prevent any of these diseases from becoming severe very quickly. Okay, so we're on to the thought questions for chapter 4. So chapter 4, thought questions. So what properties of a solute are relevant in determining the type of transport that moves the solute in and out of the cells? Okay, so the main characteristics are molecular size and hydrophobicity or hydrophilicity. If a substance is relatively small with a hydrophobic nonpolar chemical structure, then it can simply diffuse across the cell membrane's lipid core down a concentration gradient. Simple diffusion moves solutes from an area of high concentration to an area of lower concentration until an equilibrium is reached. Hydrophilic compounds require some form of membrane transport protein to move across the membrane. When cells have a receptor for a specific solute, then that solute can be taken into a cell via receptor-mediated endocytosis. Active transport moves solutes across the cell membrane against the concentration gradient. And we're going to get into that a little more in a few slides. I have a video I'm going to show you to really reinforce that. Okay. Explain the role of the sodium and potassium pump in enabling the function of secondary active transport proteins. The sodium-potassium pump moves both sodium and potassium against their concentration gradients using active transport. This creates the difference between intracellular and extracellular fluids. In secondary active transport, the downhill movement of sodium back into the cell down its concentration gradient provides energy that is used to move other solutes against their concentration gradients. This is called secondary active transport because it is energy requiring or active, but the energy is provided as a secondary effect of the sodium gradient created by the sodium-potassium pump. Number 3, what role do potassium leak channels play in creating a negative resting membrane potentials? Ion channels are pores that open to allow ions to move in or out of a cell down the concentration gradient. Most cell membranes have potassium leak channels that allow potassium to freely diffuse across the membrane. Since potassium has high intracellular concentration and low extracellular concentration, the net movement of potassium is in the outward direction. Potassium diffuses out of the cell, but negative charges are left behind since there is no mechanism for them to move out. Eventually, the intracellular negativity attracts extracellular positive charges, and an electrical potential difference is created with inside of the cell negative to outside. This negative membrane potential then attracts potassium back into the cell, with the final membrane potential determined by the balance of the electrical pull on potassium with the outward push of the potassium chemical gradient. The resting membrane potential then is close to the nearness potential for potassium movement. Number 4, page 97. Trace the events involved in signaling by GPCRs that act through the adenyl cyclase system. So an example of this signaling occurs when norepinephrine binds to an adrenergic receptor. The receptor changes shape, which increases its affinity for GS protein. The G protein is made up of alpha, beta, and Y components. When the norepinephrine binds to the receptor, the G protein dissociates into alpha and beta proteins. The A stimulate adenyl cyclase to increase its activity, increasing intracellular cyclic adenosine monophosphate. Next, those molecules activate protein kinase A, which adds a phosphate group onto target proteins, further amplifying the signal, resulting in changes of the cell function. For example, in the heart, activation of B adrenergic receptors results in calcium channel phosphorylation and other adaptations that increase heart rate and the force of contraction. Okay, the 3 breaks that terminate downstream actions of G protein coupled receptors, so GPCRs. So ligands diffuse away from the receptor, enabling receptor signaling. GTP turn off their activity until a new hormone molecule binds to the receptor and initiates G protein activation again. Phosphate enzyme remove phosphates added by protein kinases. Number 6, what are the major points of difference between tyrosine kinase linked intracellular actions and downstream actions of GPCR signaling? Tyrosine kinase is an enzyme subclass of protein kinase with the property that it phosphorylates tyrosine residues within target proteins. Most of the other protein kinases described in the chapter are serine and threonine kinases that phosphorylate serine or threonine residues in target proteins. Tyrosine kinase, when self-activated by other tyrosine kinase protein, builds up a complex of proteins in intracellular portions of receptors. In adults, the action of these receptors are brief and specific to wound healing and immune responses. Due to their potent ability to stimulate cell division, dysfunction of tyrosine kinase linked receptors often results in cancer, and tyrosine kinase blocking drugs like imatinib are used to treat many cancers. Okay, let's see if we can finish these. Okay, so number 7, how do the mechanisms of muscle contraction differ between striated and smooth muscle cells? In striated muscle cells, the sarcomere is the functional unit of muscle contraction, and the bundles of actin and myosin organized in sarcomeres are the source of striations visible in microscopic images of muscle cells. Muscle contraction occurs as myosin heads bind to actin strands and pull those strands towards the center of the sarcomere, which shortens the muscle fiber and ultimately the entire muscle. In smooth muscle cells, contraction or relaxation is controlled by the autonomic nervous system and chemical mediators. They do not have striations because they do not have sarcomeres. Instead, the actin and myosin filaments are loosely arranged, but anchored to dense bodies within the cytoplasm. Cross-bridge cycling still shortens the cell, but the force is less than what is produced by striated muscles. This means that the ATP requirements of smooth muscle cells is less than what is required by striated muscle cells, because smooth muscle requires less energy. The role of calcium also differs between the two cells. In striated muscle cells, calcium binds to troponin, shifting the myosin fibers and uncovering myosin binding sites on action, allowing cross-bridge formation. In smooth muscle cells, a calcium binds to calmodulin and activates myosin light chain kinase. Phosphorylation of myosin allows cross-bridge formation. Number 8, what are examples of these types of cells and their function? Well, we just talked about that. Skeletal muscle fibers are found in muscle cells, cardiac cells are found in the heart, and smooth muscle cells are found in walls of hollow organs such as the GI tract, bladder, and uterus. Page 106, what are the hallmarks of necrotic cell death? So, sorry guys, let's come back. I'm gonna go over that slide in just a minute. Let's just finish these review questions. We'll get into it a little more in detail. I just wanted you guys really to have the answers to these thought questions just to solidify your readings, because it's a lot of information. So necrotic cell death generally involves an appreciable number of cells. Acute ischemia, seen in myocardial infarction or a heart attack, and stroke causes death of cells at the heart of the ischemic region, where with some sparing of cells on the border that may receive blood flow from nearby collateral vessels. In the core, cells cannot make ATP, so the sodium-potassium pump cannot function. The cell accumulates sodium, causing it to swell and burst. Lysosomes burst, releasing degradative enzymes that start to digest the tissue. Loss of cell calcium homeostasis allows calcium to build up and activate calcium dependent degradative enzymes, including proteases that digest proteins. And then you have phospholipases that destroy membranes and nucleases that destroy nucleic acids. So the cell really has its own disposable way of getting rid of bad cells or cells that are dying. Mitochondria and ribosomes cease normal function, and damaged signals attract macrophages and neutrophils, creating an inflammatory focus. Why is it so important for cell death programs to exist? Like, how do they contribute to normal physiology and prevention of disease? Well, cell death or apoptosis is important during prenatal development to achieve proper shape, particularly of the digits or your fingers. Postnatally, apoptosis is important in immune cell development. Cells maturing in the thymus are assessing for their reactivity, while lymphocytes that can react to self-antigens are killed by apoptosis. This is a natural mechanism to reduce the incidence of autoimmune disease. All right, let's get into this. So here we have a comparison of the three types of muscle cells: skeletal, cardiac, and smooth. Remember, cardiac was the heart, skeletal is the skeletal system, obviously, and then the smooth muscle is the walls of the viscera, blood vessels, iris of the eye, attractor muscles of hair follicles. So this is the difference between their nuclei in each cell. So the skeletal muscle has multiple nuclei adjacent to a sarcoplasma. The cardiac only usually has one nucleus near the middle of the cell, and the smooth muscle also one nucleus near the middle of the cell. Ooh, sorry, guys. Okay. So next I wanted to get into the structure of the cell components. Okay, so here we have the cell, and look at every little piece to this cell. It's tremendous. This goes back to, undergrad for sure, but I just felt it was important to put a diagram up there so you could just review what each function of the cell is and what component it is. Okay. Good. Next, we're talking about mitosis. Now, we went into mitosis a little bit earlier, but here again is the process of nuclear division in eukaryotic cells that occurs when a parent cell divides to produce two identical daughter cells. It's conventionally divided into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. And here it is showing interphase, prophase, all of your different phases. Sometimes these visuals, and I would encourage you to look at your diagrams and your charts in your book, in your text, to really help solidify some of these concepts. Because I think visually it's much actually easier sometimes to grasp the complexity of the breakdown of this, each function of each thing in the cell. Okay, so here's a diagram of the necrotic cell death, which we just spoke about. Remember, necrotic cell death is caused by severe traumatic or ischemic injury, all right? Whereas apoptotic cell death is sometimes psychological or physiological rather than pathological. An autophagic cell death is slower, and autophagy itself may be a protective mechanism that maintains the cell and prevents cell death. Okay, so here's the two different types of cell deaths. Remember, there's necrosis, which is an uncontrolled cell death, and apoptosis, which is what they call programmed cell suicide. But it's physiological, so it's just your body is doing it to protect itself. Unlike necrosis, which is like an emergent thing that just randomly happens. So the biological cause of aging has not been definitively determined, but humans and other organisms experience gradual losses of cell and organ function that ends in death, even in the absence of a specific disease process. Different pathways can contribute to aging, including lifelong exposure to injurious chemicals such as reactive oxygen species and inflammatory cytokines, and gradual diminishment of cellular quality control systems for integrity of DNA, protein, and mitochondria. Nutrient signaling cascades appear to play a role in aging, and caloric restriction extends life in many animal models. So that's interesting. Okay, so we have a few more questions for review. So number 11, what are the top 3 classes of cellular biomolecules that are vulnerable to reactive oxygen species or ROS mediated damage? So the 3 main targets of ROS mediated damage are cell membranes that undergo lipid peroxidation, proteins that undergo oxidative modification, and DNA that can undergo breaks and strand cross-linking. Number 12, explain the role of telomeres in maintaining chromosomal integrity over repeated cycles of mitosis. So telomeres are necessary for complete replication of the DNA during mitosis. The telomeres grow progressively shorter with each round of replication. Eventually, the telomere is too short and is flagged as damaged, causing cell arrest. Somatic cells lack telomerase, an enzyme that can extend telomere length. So these cells reach a point of no return, after which they can no longer replicate the DNA and produce daughter cells. Okay, so I really wanted to give you a nice little review on the mechanisms of cell membrane transport, and I just found this video to be helpful and not too long, so it can actually keep your focus. So just let's watch this review.
Membrane transport. So how do substances move into and out of a cell? Well, cells have a plasma membrane that acts like a fence, and this plasma membrane is what regulates what moves into and out of a cell. The membrane is comprised mainly of phospholipid molecules and has protein molecules scattered throughout its structure. The phospholipid molecules have a polar hydrophilic water-loving head and 2 nonpolar hydrophobic water-fearing tails. The membrane is formed as the heads face out and the tails face in, forming a bilayer, as you can see here. The inner part of the cell, called the cytosol, is here, and the outside of the cell is surrounded by a fluid called interstitial fluid. Substances move into and out of a cell through several different processes called membrane transport. There are 2 main processes: passive transport processes and active transport processes. The main difference between the 2 is that passive processes do not require energy expenditure, and active processes do require the cells to expend energy. Let's start by looking at the passive processes, which include simple diffusion, facilitated diffusion, and osmosis. Diffusion is the movement of a substance from where it has a high concentration to where it has a low concentration, or the tendency of a substance to spread out evenly over a given space. For instance, when a sugar cube is dissolved in water, over time the cube will dissolve and eventually spread out evenly in the water until it reaches equilibrium. Diffusion occurs down a concentration gradient, which is a difference in concentration of a substance between 2 areas. So the sugar molecules will move from an area of high concentration to an area of low concentration. Cellular diffusion is when diffusion of a solute, which is a dissolved substance, occurs across the plasma membrane from an area of high concentration to an area of low concentration. This is dependent on the concentration of a substance in the interstitial fluid outside of the cell and the cytosol inside of the cell. This can occur through simple diffusion or facilitated diffusion. Simple diffusion occurs with solutes that are small and nonpolar. By being nonpolar, they can move in between phospholipid molecules that form the plasma membrane because the interior region of the membrane is nonpolar. Some of the materials that move by simple diffusion include the gases oxygen and carbon dioxide and small fatty acids. So if there's a higher concentration of oxygen O2 molecules outside of a cell, they can move down the concentration gradient across the membrane without assistance and into the cell as long as the concentration gradient exists. And if there's a higher concentration of carbon dioxide, CO2 molecules inside a cell, they can move across the membrane without assistance, out of the cell into the interstitial fluid. So again, simple diffusion is when nonpolar molecules pass directly through a membrane. The second type of diffusion is facilitated diffusion. This applies to solutes that are small and either charged or polar. Because these solutes are polar, the nonpolar phospholipid bilayer blocks them from passing through the membrane and into or out of the cell by simple diffusion. However, they can pass into and out of the cell with the assistance of plasma membrane proteins through a process called facilitated diffusion. There are two types of facilitated diffusion: channel-mediated diffusion and carrier-mediated diffusion. The difference between the two is the type of transport protein used to move the substance across the membrane. Channel-mediated diffusion is when an ion, which is a charged particle where its total number of electrons does not equal its total number of protons, giving it a positive or negative charge, moves across the membrane through a water-filled protein channel. Each protein channel is typically specific for one type of ion, and there are two types of channels: a leak channel, which is continuously open, and a gated channel, which only opens due to a stimulus and only stays open for a fraction of a second. So for a sodium positive ion, it can pass through a sodium positive leak channel continuously, and a gated sodium positive channel will only open due to a stimulus to allow the ion to pass through into the cell. Carrier-mediated diffusion involves the movement of polar molecules, such as simple sugars or simple carbohydrates, and amino acids across the membrane. This is accomplished by a carrier protein, which actually changes shape in the process. For instance, glucose binds to a carrier protein, which changes shape and moves the glucose molecule to the other side of the membrane. Now for osmosis. Osmosis is the passive movement of water through a selectively permeable membrane.
It's 6 o'clock.
This occurs when there's a difference in concentration of water on either side of the membrane. This can happen in one of two ways. Water can slip between the phospholipid molecules that make up the plasma membrane, or through integral protein water channels that are called aquaporins. The plasma membrane is not permeable to most solutes, such as charged, polar, and large substances. So for example, one side of the membrane, the cytosol or interstitial fluid, can have more solutes than the other side. Let's say the interstitial fluid has 3% solutes and 97% water, and the cytosol side has 1% solutes and 99% water. In this example, water will move down its concentration gradient from the 99% cytosol side to the 97% interstitial side to achieve equilibrium. Now let's look at active processes. Active processes require the use of cellular energy for membrane transport. There are two types of active processes: active transport and vesicular transport. Active transport is the movement of a solute against its concentration gradient, or going from an area of low concentration to a place of higher concentration. Vesicular transport is the transport of large substances across the plasma membrane by a vesicule, which is a membrane-bound sac filled with materials. Active transport has two types: primary active transport and secondary active transport. In primary active transport, cellular protein pumps, called ion pumps, move ions across the membrane against their concentration gradient. Let's take a look at an example. Here we have a membrane along with a sodium positive and potassium positive protein pump. 3 sodium ions and ATP bind to the pump, ATP being like fuel or the form of cellular energy. In order to release that energy, ATP breaks down into ADP and P. P binds to the pump, and the release of energy causes the pump to change shape and release the sodium positive ions into the interstitial fluid. 2 positive potassium ions bind to the pump, and the P produced by the ATP is released into the cytosol of the cell. The transport protein, or pump, reverts back to its original shape, releasing the positive potassium ions into the cytosol, and the pump is ready to do its job again. In secondary active transport, a substance is moved against its concentration gradient by using energy provided by the movement of a second substance down its concentration gradient. So a substance that is moving from a place of high concentration to low concentration will provide the energy to move a second substance from a place of low concentration to high concentration. There are 2 types of secondary active transports: symport, where 2 substances are moved in the same direction, and antiport, where 2 substances are moved in opposite directions. In our example here, we have positive sodium ions moving from high concentration outside of the cell to lower concentration inside of the cell. A symporter will use the energy created by this to move a glucose molecule from a low concentration area to a high concentration area. And here we have an antiporter, again using the energy from moving positive sodium ions from high concentration outside of the cell to lower concentration inside of the cell, and H positive is moved up its gradient and outside of the cell. Vesicular transport involves the transport of larger substances, such as proteins or large carbohydrate polysaccharides, across the plasma membrane. There are two types of vesicular transport, exocytosis and endocytosis. In exocytosis, materials are secreted from the cell to the interstitial fluid outside of the cell. Typically, membranous vesicles formed by the Golgi apparatus transport materials to the cell's membrane. At the membrane, the phospholipid molecules that make up the vesicle fuse with the phospholipid molecules that make up the plasma membrane, and the contents of the vesicle are released outside of the cell. The vesicle membrane now becomes part of the plasma membrane. In endocytosis, the plasma membrane kind of traps the substance by folding inward. The lipid bilayer then fuses to form a vesicle surrounding the substance. There are three main forms of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. In phagocytosis, a large particle is engulfed by the newly formed vesicle, and this vesicle fuses with a lysosome, which is a membranous vesicle that contains digestive enzymes that break down the particle into its component molecules. Phagocytosis is also called cell eating. Pinocytosis, which is also referred to as cell drinking, is when the plasma membrane folds inward and engulfs droplets of interstitial fluid that contain dissolved solutes that can be used by the cell. Receptor-mediated endocytosis involves using receptors on the outside of the plasma membrane. These receptors bind with molecules in the interstitial fluid, and the membrane folds, enclosing the receptors and the bound molecules to form a vesicle for transport within the cell. One last thing to note: both vesicular transport processes, endocytosis and exocytosis, require energy. And that be the basics on transport across a membrane.
Okay, I hope this helped you guys just to give you a kind of a broad overview. And if you notice, this guy actually has some other videos that would probably be super helpful for you guys if you wanted to have more reviews that you could look into if you wanted. Okay, so mechanisms of cell signaling. So another quick video just to kind of summarize like how the cells are communicating to each other and their pathways that they use to do so.
To make a multicellular organism, cells must be able to communicate with one another. And to do it, cells often send out tiny chemical signals that act on the receptors of other cells. Signals can be classified according to the distance between the signaling cell and the target cell. Autocrine signals are produced by a cell and go to its own receptors, so the cell sends a signal to itself. Paracrine signals are produced by a cell and go to target cells that are nearby. And endocrine signals are produced by a cell and go to target cells that are further away. Examples of these include hormones that are secreted into the bloodstream, as well as cytokines that can be released at the site of injury and act on the brain to cause a fever. Signaling molecules, or ligands, can be hydrophobic, meaning that they tend to repel water, or hydrophilic, meaning that they tend to stay in water. Hydrophobic signaling molecules can't freely float in the extracellular space, so they're brought into the target cells by carrier proteins. Hydrophobic molecules can diffuse across the cell membrane and bind to receptor proteins inside the target cell, either in the cytoplasm or in the nucleus. Most signal molecules are hydrophilic, so they can freely float in the extracellular space to reach the target cells, but are then unable to cross the cell membrane. So to pass on the signal, hydrophilic molecules bind in receptors on the cell surface. These receptors are transmembrane proteins with an extracellular end that binds to the ligand and an intracellular end that triggers a signaling pathway inside the cell.
We can think of the cell signaling pathway in three stages. The first stage is reception, which is when the target cell's receptors bind to a ligand. It's like a key fitting into a lock. Then there's transduction, which means that the receptor protein changes in some way, and that activates intracellular molecules, the second messengers. The third stage is the cell's response to the signal. Zooming into these transmembrane receptors, there are three major classes: G protein coupled receptors, enzyme coupled receptors, and ion channel receptors. G protein coupled receptors are 7-pass transmembrane receptors. These are like really long proteins that have one end that sits outside the cell and binds the ligand. Then the snake-like protein dips in and out of the cell membrane 7 times and finally ends on the inside of the cell. The end of the G protein coupled receptor that's within the cell activates intracellular proteins called guanine nucleotide binding proteins, or G proteins. G proteins are made up of 3 subunits called alpha, beta, and gamma, sort of like a flower with 3 petals. The alpha and the gamma subunits are anchored to the cell membrane and keep the G protein right next to the receptor. G proteins bind to guanosine diphosphate, or GDP, when they're inactive. When the alpha subunit is bound to GDP, the 3 subunits stay together so the flower is closed. But when the ligand binds, the G protein coupled receptor changes its shape, and this allows the G protein to release GDP and bind GTP instead, activating the protein. When the alpha subunit is bound to GTP, the alpha subunit separates from the beta and gamma subunits, like one petal opening and separating from the others. When that happens, the alpha subunit is free to interact with other proteins. It stimulates some while inhibiting others. But to act on other proteins, the alpha subunit turns GTP into GDP, and when that happens, the 3 subunits come together again. The flower closes, and the G protein is turned off. Overall, there are 3 types of G proteins: GQ, GI, and GS, and each one stimulates and inhibits a different set of enzymes and molecular pathways. The GQ protein activates the enzyme phospholipase C, which is found in the cell membrane. Phospholipase C then cleaves a phospholipid called phosphatidylinositol 4,5-bisphosphate into inositol trisphosphate and diacylglycerol. Inositol trisphosphate is soluble and diffuses freely through the cytoplasm and into the endoplasmic reticulum, where it opens up calcium channels. Since the calcium concentration is higher in the endoplasmic reticulum than in the cytoplasm, calcium flows out of the endoplasmic reticulum to the cytoplasm. The increased calcium concentration in the cytoplasm changes the electrical charge of the cell and can lead to depolarization. Meanwhile, diacylglycerol remains attached to the cell membrane and binds to the enzyme protein kinase C, which also relies on calcium to fully activate. Once calcium levels in the cell go up, protein kinase C starts to activate proteins by adding phosphoryl groups to them. Next is protein GS, which stimulates the enzyme adenylate cyclase. Activated adenylate cyclase takes adenosine triphosphate, or ATP, and removes 2 phosphate molecules, transforming it into cyclic adenosine monophosphate, or CAMP. CAMP moves throughout the cytoplasm and binds to the enzyme protein kinase A. Protein kinase A has 2 parts, a regulatory subunit and a catalytic subunit, and CAMP specifically binds the regulatory subunit of protein kinase A. When CAMP binds, it makes the regulatory subunit dissociate from the catalytic subunit. It's kind of like pulling the pin out of a fire extinguisher, allowing it, in this case it being the catalytic subunit, to do its job. So after dissociating, the catalytic subunit of protein kinase A is free to phosphorylate target proteins that trigger a cellular response. Finally, there's the protein GI, which is also bound to adenylate cyclase, but in this case, it inhibits it, causing negative feedback on protein GS. This is particularly important in helping to inactivate cells. Next are the enzyme-coupled receptors. They're usually single-pass transmembrane proteins, meaning that they have only one transmembrane segment, and their intracellular end has intrinsic enzyme activity. In other words, enzyme-coupled receptors have 2 parts. One domain is the receptor, and the other domain is an enzyme. Each domain has a separate function, like on a Swiss Army knife that's composed of both a knife and scissors. The enzymatic domain is usually a protein kinase that phosphorylates the receptor domain. Now, there are three main types of enzyme-coupled receptors based on the amino acid at which the receptors get phosphorylated. The first group are the receptor tyrosine kinases. These are the most common enzyme-coupled receptors, and there are many subfamilies. Receptor tyrosine kinase are generally molecules that can't phosphorylate their own tyrosine side chains. When a ligand binds, two receptor chains come together and dimerize, and they cross-phosphorylate one another at multiple tyrosine residues. This triggers a conformational change that creates high-affinity binding sites for the second messengers, which can also be phosphorylated and activated, triggering the signaling pathway. Next are the tyrosine kinase-associated receptors, which work in nearly the same way as receptor tyrosine kinases, and their name even sounds almost the same. The key difference is that they have no intrinsic enzyme activity. Instead, they're associated with cytoplasmic tyrosine kinases. When the receptors bind their ligand, the cytoplasmic tyrosine kinases phosphorylate various target proteins to relay the signal. Finally, there are the receptor serine-threonine kinases, and they have a serine-threonine kinase domain on their intracellular end. There are two classes of these receptor serine-threonine kinases, type 1 and type 2, which are structurally similar. Ligand binding brings the two together so that the type 2 receptor can phosphorylate and activate the type 1 receptor, which in turn recruits and phosphorylates various target proteins to relay the signal. Finally, there are the ion channel receptors, which are generally closed, but then they open up once they bind a specific ligand. They allow ions like chloride, calcium, sodium, and potassium to passively flow down their gradient. This leads to a shift in electric charge distribution inside the cell, triggering a cellular response. All right, as a quick recap, autocrine signals target the same cell, paracrine signals target nearby cells, and endocrine signals target distant cells. Hydrophobic ligands are able to diffuse across the cell membrane and bind to receptor proteins inside the target cells. Hydrophilic ligands are unable to cross the cell membrane, so they must bind to transmembrane receptors, which have an intracellular end that triggers a signaling pathway inside the target cell. There are three major transmembrane receptor classes: G protein coupled receptors, enzyme coupled receptors, and ion channel receptors.
Okay, hopefully this was helpful for you guys. It was only like a 10-minute video and it gave you a lot of information with just a lot of visual explanation of the mechanisms of cell signaling. Okay, so the last thing in this chapter that, I definitely wanted to hone in on and make sure you guys are aware of, it's a little less complicated in terms of trying to grasp the concept, but it is very important in terms of considering how genetics and the effect of cells are on the hallmarks of aging and what happens during the aging process when it comes to cellular function. This doctor does a great job of talking about the 9 hallmarks of aging, and it really makes you consider, how you treat your body and how we educate our patients about, how to prevent aging and what to do to slow it down and give you the most healthy options to prevent cell disease or cell death.
To age or not to age? That actually is the question. How's it going? It is Dr. V, triple board certified MD and performance and longevity expert. And on this video, I'm going to talk to you guys about the 9 different biological hallmarks of aging. Now, you might be wondering why we age. Well, what exactly is aging? What is a young person versus what is an old person? Well, typically find somebody who's younger tends to have more energy, right? They've got a youthful look to them. They look younger, right? So that's youth. So why do people age? What happens during that aging process? Now, if you read all the longevity books, they boil it down to 9 factors that happen for aging. And that's what this video is going to cover today. So from the inside out, let's take you into the cell
and all the way back out into the human organism, 9 factors that determine aging. Number 1, genomic instability. So we all know what DNA is, right? DNA makes RNA, makes protein, and those proteins kind of make up who we are as people. Over time, there are many things in our environment, including toxins, including how we live our lives, that could actually damage our DNA. And if that doesn't get repaired, again, the DNA is that blueprint. It makes RNA, it makes protein. And if you damage that DNA, well, we're going to get the wrong protein. We're going to get the wrong signals. So if your DNA starts to become unstable, and over time, our ability to protect its stability decreases. So genomic instability is one of the hallmarks of aging. What's number 2? Number 2 is telomere attrition. What are telomeres? Telomeres are these base pairs at the end of the DNA. Basically, they're just the end caps of our DNA that protects our DNA from aging. But over time and with stress, these telomeres get shorter and shorter and shorter. And as they shorten, they also shorten the life of a cell. So telomere attrition is hallmark number 2 of aging. Hallmark number 3 of aging is epigenetic modification. What is epigenetics? Well, it's what we talk about in our book, Thrive State. And hey, if you haven't picked up a copy yet, go ahead and pick up a copy of thrivestatebook.com, where I talk about how the things that we experience in life, our emotions, our thoughts, how we live our lives, how we show up every day, actually changes how our DNA is being expressed. And it does so because depending on how we live our life, we actually create an energetic system that actually starts to mix these little marks on our DNA. These are epigenetic tags. And these epigenetic tags actually tell us how old we become. So over time, with our experiences and how we live our lives, we start to add these things into our DNA, all these epigenetic tags. And that is another hallmark of aging. Number 3, epigenetic tags. Number 4 hallmark of aging is protein instability. So as we get a little bit older, proteins basically make up what we are, right? We've got collagen protein in our skin that keeps us, makes our skin feel nice and firm. As we start to lose that, or as the collagen breaks down, we start to get wrinkles and things like that. So proteins are the building blocks of who we are. Proteins are the building blocks of life. And when they start to become unstable and they don't do their job very well, guess what? Life doesn't go on very well. So another hallmark of aging is that proteins start to function very well. Protein instability, another hallmark of aging. All right, so far we've talked about the things in the nucleus when it comes to aging. Our DNA, right? Our genomic instability. Number 2, that those telomeres at the ends of the DNA. And number 3, we would talk about those epigenetic tags to our DNA. Then we talked about things outside of our DNA, such as the proteins, they become unstable. Now, let's move to the mitochondria. Mitochondrial dysfunction is another factor of cells aging. What do the mitochondria do? Well, mitochondria are actually the power plants of all our cells. And over time, because of free radical damage, because of how we live our lives, we could have mitochondrial dysfunction. Imagine, any device that you have. You know what happens when your device, when you're low in battery in any one of those devices, it just doesn't work as well, right? The same thing happens with our cells. When those mitochondria don't work very well, imagine that cell not having enough energy to do what it's supposed to do. Well, guess what? That thing is going to function at a lower level. So number 5, hallmark of aging is mitochondrial dysfunction. So what is another hallmark of aging? Well, nutrient sensing actually becomes impaired. When we are young, we are able to take in a lot of information like light, like sound, like all these nutrients that come into our body. We're able to sense protein, fats, ATP, all of these nutrient sensing capabilities that allow us to be able to know what's going on in the environment. As we age, that nutrient sensing capability goes down. So poor nutrient sensing is also another hallmark of aging. The next hallmark of aging is poor intercellular communication. We are a multi-cellular organism. We've got lots of different cells in our own body. Then we've got tons of microbes that actually live in our body as well. And it's an intricate dance and an intricate communication of all these things happening that make us who we are. And cells have to be able to talk to each other very well. For example, if there was a virus that entered your body, once that virus gets detected, our immune system has to raise the red flag and signal to the rest of the body, hey, there is a foreign invader here. We have to mount an inflammatory response. And that communication system has to turn on and has to go right in order to save that person from that invader. So intracellular communication is so important. As we age, it seems that communication breaks down. The next hallmark of aging is cellular senescence. That means cells are getting old. Cells are actually turning into zombies. What happens here? Well, you build up a lot of junk, and these cells build up a lot of junk. They don't break down the junk, and the cell can't divide anymore, and it's just sitting around. All right, they're known as zombie cells. In fact, these zombie cells actually send out signals that causes inflammation to happen. So as we age, we tend to create these cells that don't do very much anymore, but also damaging to the cells around it. That's called cellular senescence, another hallmark of aging. And the last hallmark of aging is something called stem cell attrition. You know, when babies are born, there's actually stem cells in the umbilical cord, in the Gordon's jelly, all around the blood vessels of the baby are these stem cells. We actually have these stem cells now as well. They tend to age and they will go through a process of attrition, meaning they don't work as well. Why are stem cells important? Well, anytime we get injured, anytime something happens where there's inflammation or injury, well, stem cells get activated and will go to that spot of injury and they'll start the repair process. So stem cells are so important to help with that repair process. Well, unfortunately, as we age, so do our stem cells, and our ability to repair itself as we age, well, that doesn't happen as well either. There you have it, the 9 hallmarks of aging. So what exactly is aging? I mean, when we were very, very young as babies, our cells basically had the ability to do anything, repair against damage. It was very resilient. And over time, these 9 things happen, and we lose that resiliency to all the things that we happen to encounter, whether it be toxins, whether it be toxic emotions, and all these things that happen to us in life. And that is really the process of aging. So again, what happens in the biological level of aging? Let's review them. Things that are within the cell nucleus: genomic instability, telomere attrition, and genetic modification. Just outside the cell, at the protein level, proteins start to become unstable. Then you have mitochondrial dysfunction. Then we lose our ability to sense nutrients very well. Poor intercellular communication, stem cell attrition, and then also cellular senescence. These are the things biologically that happen. These are the 9 hallmarks of aging. Now, I tell you all these things not to scare you, but just to let you know, these are the things that are happening that lead to aging. However, if you start to read my book, Thrive State, you'll actually know the lifestyle factors that actually put the brakes on any of these things actually happening. When you master your bioenergetic state, when you master your sleep, your nutrition, your movement, your stress and emotional mastery, when you start to master your relationships, your thought and your mindset, you master your sense of purpose and showing up in the world like a vibrant being, you start to actually take control and avoid those 9 hallmarks of aging that you see happening. Because remember always, you are your best medicine.
Okay, that was an awesome video. For me, that was my favorite video that I have seen or shown in any of my lectures, because he really puts it in practical terms on how to apply all of these things at these cellular levels to real life, and how it applies to us, you know, from a level that we can actually understand it and apply to our life going forward. I actually would really love to read his book, because I think it would probably put all of this into perspective. Okay, so aging at a cellular level. So this is a great diagram because it talks about all those aging effects that he just went through, but it also talks about, you know, how physical exercise can actually help prevent these aging effects, and the importance of even teaching our geriatric patients what exercise can do, whether it's a simple walk, whether it's chair yoga. These are the kinds of things that you now will understand because you understand things at a cellular level, which is what you're learning and why you're learning it, so that you can really understand the depth of the importance of teaching your patients and educating them across the lifespan on the importance of, at a multi-system level, on the importance of teaching them all these different factors and how to apply it to their life and encourage health promotion and encourage primary prevention in your plan of care. All right, guys, thank you so much. I hope this really gave you a broad understanding and kind of outline of your readings. And next week, we'll be focusing on chapter 7 with neoplasia. All right, see you guys next week. Thank you.